Uh, Good morning to everybody. Torsten, I'm on, right?
You're on, yes.
Yeah. So today we also actually welcome members of summer school, the Motif Summer School of Fibrosis, which was originally planned for last year before the pandemic hit. And this is actually for students in Bergen, Toronto, and San Francisco. And due to the time difference, I don't expect too many of them to be up 3 o'clock in the morning to listen to this first lecture, but they will have access to the videos. And we hope that some of them will join later in the afternoon. And today also, one of today's speakers, Sergei Plotnikov, will be with us and share a practical seminar about traction force microscopy, which we look forward to. So from now on till the end, we're both BMed 904 and the summer school for fibrosis with members from Toronto and San Francisco, and we're very glad to have them with us. Okay, that was that, and then we go on to today's lecture. So in the next coming days, we will talk. There will be several talk about different aspects of fibrosis, which is the synthesis of extracellular matrix leading to pathological changes. And so I decided I will kind of give an introductory lecture to fibroblasts. And what I have done today is more collecting references. So you will see throughout the lecture first pages of recent good articles that you can read for further understanding. And I will not have time during this lecture to go in that much detail. So what I will talk about today is that we will say a few words about what is a fibroblast, and related to that, what is a myofibroblast and what is a cancer-associated fibroblast, also called CAF. Very relevant to this is also where do fibroblasts come from? And in that connection, we will say a few words about cell lineage tracing. And then we will say a few words about what do fibroblasts do. And in that context, heterogeneity comes into the picture. When you talk about fibroblasts, is very dangerous to generalize because they're different type of fibroblast, even within a tissue at a particular time. We will say a few words about introductory words about fibrosis. You will hear more about that, and you will hear much more during these next few days about cancer-associated fibroblasts. So I will just say a few words and hint you towards good recent articles. So we start with how you define this cell type. So you might think that these cells that have been described for more than 100 years, fibroblasts, by now should be well defined, but actually they are not. So to say what is a fibroblast, or to answer what is a fibroblast, is not easy at all. The standard description that you will find in many textbooks is that it is a connective tissue cell that produce extracellular matrix. And often it will also say that these cells are spindle shaped, like this picture, or stellate shaped. So that's morphology, how you can detect them if you look at the histological section. So for a long time, one used markers related to collagen synthesis to describe fibroblast in a tissue. And one of the enzymes that we talked about in the ECM lecture was prolyl hydroxylase, a specific enzyme involving collagen biosynthesis. And that was often used as a marker for fibroblast in tissues. So bottom line is it's difficult to define fibroblast. And one reason that it is so difficult is that there are no good biomarker, no good antigen that is present on fibroblasts only. So you cannot take one antibody and say, I'm going to stain for fibroblasts. Most available reagents will react with different cell types. Another problem that we will come to is that fibroblasts are so heterogeneous, they in different aspects. So this is from a review we wrote some time ago, this definition. And already after this paper was published, things have happened that have changed things. And we will come to that later in the article. Super good review was published. Now let me find the pointer. In 2020, which is a status of the field for cancer-associated fibroblasts, where first author is Eric Sahai from London, who was here in Bergen on another course that we gave some time ago, and senior author is now the late Sina Werb from UCSF. And these are various experts on fibroblasts that have written one document that has summarized the state of the art. And we will look at some of the pictures from this article that I can strongly recommend. But in the definition of what is a fibroblast in this paper, they say it starts with the definition of a fibroblast is surprisingly tricky. And then they go on to say something that is very important, something that we have learned in recent years, that the embryonic origin of most fibroblasts is from the primary mesenchyme that develops from the mesoderm, and a small subset of fibroblasts comes from the neural crest, which comes from the ectoderm. So that is a fact. Fibroblasts embryonically come from the mesoderm or are derived from neural crest. And then they go on to, since there are no specific marker, you often have to use a combination of markers. And these are some of the markers you will hear about in the next few days. You already heard from Kalle Sippler talk about PDF alpha receptor being a universal fibroblast marker in the skin. but that doesn't apply to all tissues. So this varies from tissue to tissue. So related to what is a fibroblast is of course also what is a myofibroblast. So the basic question of how to define a fibroblastic cell, that problem persists also for myofibroblast. But myofibroblast is an activated fibroblast. And more now we're starting to understand that the activation of a myofibroblast can mean different things. Often, traditionally, activation means that they become contractile. And in order to be contractile and contract the tissue, they express a special actin isoform that's called smooth muscle actin that makes the cells extra contractile. But more and more, we're starting to realize that activation can mean different things. It not necessarily only increased contractility, it can also mean other things. And so fibroblast activation is not just increase in contractility, it can also mean other things. And the cell type that we will talk a lot about in the coming days is the cancer-associated fibroblast. So these are the cells that surround the tumor epithelial cells, the tumor cells. And again, this basic question of what is a fibroblast persists, but in addition, there is increased heterogeneity to cancer-associated fibroblasts because now there are many more sources of cancer-associated fibroblast. And I would say over the past 10 years, there's been different favorite sources of CAFs. So for a while, it was very fancy and novel to say that the CAFs came from the circulation, fibrocytes in the circulation that home to the tumor. For another period, it was very popular to say that, or to publish that fibroblasts came from EMT, epithelial mesenchymal transition. But at the end of the day, when we know more, thanks to new techniques, what we're landing in is that a majority of the CAFs come from endogenous fibroblasts in the tissue. The exception is that in some tissues we don't have any fibroblast, and that is, or not much fibroblast, but we have more of a cell type that's associated with blood vessels called pericytes. So that's the case in the liver. There are not very many fibroblasts, except those associated with large blood vessels. So if you have a liver tumor, then it's the pericytes or the stellate cells that will become activated and that become the CAFs. The same in the pancreas. There is a lot of stellate cells in the pancreas. And when you have a tumor, pancreatic tumor, it is these cells that become activated and form the CAFs. So again, back to this consensus statement from Eric Sahai and Sina Werb. They summarize what I just said, that CAFs, just as for fibroblast, are characterized by what they here call lineage exclusion, that you say it's cells that are not epithelial, not endothelial, not perivascular, and the stromal cells that you remain with are then the CAFs. So for all these categories, fibroblast, myofibroblast, cancer-associated fibroblasts, the definition is a problem. And many of the markers that I will not have time to go into are expressed by other cells. So the number one classical marker in addition to prolyl hydroxylase is vimentin, which is the intermediate filament protein that is expressed in fibroblasts, but it's also expressed in other cell types. And as we said, the number one marker for myofibroblast, when we consider the activation of contractility, is smooth muscle actin, alpha SMA. Okay, so let's see. So I said in this consensus statement, I think we have to turn off videos, otherwise this will show up in the recording. Margarete, that's strange, but that's how it is.
Is it a problem if you see me on the video?
No, it can be, it's just distracting if there are several, but I think the square is still there, yeah. Okay, so then origin of fibroblasts. So we said already in this consensus statement that fibroblast, we now know with more certainty where they come from. And this is due to new techniques. The problem before we had this novel technique like cell lineage tracing was that many of the reagents that was used to detect fibroblasts were not specific. So the antibodies used were not super specific. So there are many papers published where one has reached the wrong conclusion due to poor antibodies. But now there are new techniques. So one technique that enables you to look at the origin of fibroblast in experimental animals is cell lineage tracing, so that you can use in mouse. So in humans, of course, you cannot do these experimental techniques. There you have to rely still on antibodies. So Kalle Sippeli from London already talked about this, that with what we call Cree driver strain, where you have the promoter of a certain gene, and you let breed these mice with reporter mice, where you have a flux, where you have a reporter gene that becomes activated. We have done this for the integrin alpha 11, and here we see beta galactosidase staining in a developing embryo in fibroblast in an embryo. So this technique, cell lineage tracing, has enabled us to with greater certainty say where fibroblasts come from. So, as we already said then, the cell lineage tracing has established that fibroblasts, however we chose to define them, come from the mesoderm and the neural crest. So the mesoderm, that's the majority of the fibroblasts in the body. They come from the germ layer mesoderm. Neural crest comes from the ectoderm and give rise to specialized fibroblasts, especially in the head, but also in specific regions, the valves of the heart, and specific regions in the intestine. And so it's a minority of cases, but these are some fibroblasts come. So a term that you will hear a lot in this context when we talk about fibroblasts is mesoderm and mesenchyme. So what's the difference? So in this slide, you have a summary of what's the difference. So mesoderm is a germ layer that only exists in the embryo. So if you talk early during embryogenesis, you have the mesoderm, but soon after gastrulation and afterwards, mesoderms becomes other cell types. So then it's mesodermally derived. And mesenchyme is defined as a part of mesoderm of this embryonic germ layer, which develops into connective tissue and give rise to cartilage, bone, and connective tissues. And these cells, so a cell that you have in the adult that is a fibroblast, that's a mesenchymal cell. And less often you can say it's mesodermally derived, because mesoderm exists only during embryogenesis and early during embryogenesis. So that's just for information to keep the right nomenclature. And this is a picture of the different germ layers, and often in textbook you will see frog embryogenesis with the mesoderm. Here is a germ layer between underlying the ectoderm and located between ectoderm and endoderm. So in this context, there is also a very good paper from last year from Valerie Lebleu and Eric Nelson. And this is a picture from their paper. Here they don't talk about neural crest derived. They talk about the mesoderm derived cells. They also mention other cells that are fibroblast-like, which are different from the fibroblast. And this picture also shows that the earlier you go in development, the more plasticity these cells have, and with time, fibroblasts lose this plasticity and cannot change into something else. Okay, that was origin of fibroblast. Now let's look at some pictures of myofibroblasts. So myofibroblasts are activated fibroblasts that express this contractile isoform of actin called smooth muscle actin. And this is a review article in Journal of Cell Science where they recently have collected all there is to know about myofibroblast. And former postdoc of Boris Hinz is coming here is talking later about myofibroblast. This is a slide showing the different sources of CAFs. And as I said, what we know nowadays is that a majority of the CAFs in almost all tissues are from the resident fibroblast. And as we said, an exception is then the liver and the pancreas, where the parasites, or in these tissues called the stellate cells, are the major source of the CAFs. And what makes, and you will hear about this in coming day, what activates a fibroblast to become a CAF then? So there are a number of signals, and just as in fibrosis, a fibroblast is activated to become myofibroblast. One major source of activation is transforming growth factor beta as a very strong driver to CAF activation. But there is also an increased focus on understanding the influence of inflammatory signals from immune cells in the tissue, where IL-1 and IL-6 are two strong signals. And there is also a great interest now in stiffness, understanding how stiffness activates fibroblasts, which in turn then the activated CAF in turn stimulates the tumor cells. And what do fibroblasts and CAFs do? This is also something that we will hear about. Well, CAFs, of course, produce extracellular matrix. And also remodel the extracellular matrix, makes the matrix suitable for invasion, for example. Via soluble factors, they also stimulate cancer invasion in various ways. They can secrete proteases that create a pathway in the microenvironment where the tumor cells can migrate. And then in much more complex ways also secrete cytokines that stimulate the cancer cells to undergo EMT or become more mesenchymal. So soluble signals, matrix remodeling enzymes. And something that is receiving an increased interest is that CAFs also talk with the immune cells and either recruit immune cells that then in turn secrete things or affect the tumor cells and can make the tumor more aggressive in the tumor microenvironment. Right. So we said that there are now new technical developments that has changed the field completely, that has made us understand these cells much better. We already mentioned cell lineage tracing for this determining origin in experimental animals. Another technique that has revolutionized this field is what Kalle also mentioned, single cell RNA-seq. So it's short for RNA sequencing. And this has then of course builds on that. You have to dissociate, if you do that on a tissue sample, on a fibrotic tissue or on a tumor or tumor tissue, you have to isolate single cells, separate them, and then you isolate RNA from the single cells and sequence and determine the transcriptional profile on a single cell basis. And then based on this, you can create plots where you create clusters of cells, where they cluster based on their transcriptional profile. So this is a picture from this paper. So you have the tissue, you make the single cells, you do the sequencing, and then you get these clusters. And in this case, they've taken a pancreatic tumor, and they see that they have different cluster subset of tumor cells, and then they can also see fibroblasts, which in themselves have different subclusters. So now you can go in and look at the fibroblast within these clusters. So this has revolutionized cell identity in tissues and in diseased tissues, such as fibrotic tissues and in tumors. So here is analysis of the human heart. And here you can see how many clusters you have. And for when it comes to fibroblasts, that's cluster 2, cluster 4, cluster 8, 2, 4, 8. All these are fibroblasts with different transcriptional profile. And this has now been done for the lung, the liver, the kidney, and one is comparing mouse and human, one is comparing normal versus fibrotic, normal versus tumors in these tissues. So, as we said, you will hear about fibrosis, which during the coming days. So fibrosis is the overproduction of the extracellular matrix, which happens in response to injury. So in a normal injury, you will have repair and things go back to normal. But when you have an injury that doesn't heal for some reason, you will have a pathological response and pathological remodeling of the extracellular matrix. So this is a normal wound healing, an early paper from an American group that could distinguish between the different fibroblasts that Kalle talked about yesterday, papillary and reticular fibroblast, and a marker called the engrailed intracellular nuclear protein that is expressed in these fibroblasts, so they can follow the fibroblasts, how they migrate into the wound. So this is the normal healing response. And after apoptosis, after completed wound healing, a very common response is that fibroblasts undergo apoptosis, and the tissue, granulation tissue, eventually assumes a more normal structure, how it was before. But in fibrosis, then you have a number of things going on. So you have, as we said, you have excessive ECM remodeling. More matrix is produced, it's tension builds up as stiffness increases, and a number of other things happen with the tissue, both regarding vascularization, and if you try to treat the tissue with some drugs, they will have problems getting to the tissue because it's so full with extracellular matrix. And this is a graphic illustration from a very good review, which compares fibrosis and cancer. And here we have zoomed in on the cell surface where we see the integrin sitting there. And here we have in a fibrotic tissue or tumor, there is much more matrix. Integrins are clustered and activated. And one central event that we already talked about when we talked about integrin was, of course, that TGF-beta is activated in an integrin-dependent process. So that's a key event in both fibrosis. So a very good review that was published last year. Talks about fibrosis in different organs and in a very broad sense. And they talk about what we have talked about, fibroblast heterogeneity, describing papers from arthritis research showing that there are different subtypes of fibroblasts in the joint. And some are destructive and some are help to counteract the inflammation. And these are expressed, characterized by some marker expression. So there is protein called fibroblast activation protein that is present on both. And Kalle talked about the cell surface marker called CD90. That's also called Thi1, which is a membrane protein that's present in the immune effector and absent in the destructive fibroblast. They also mention characterization of fibroblast in different types of muscle, which can either work to promote matrix or not promote matrix. They also mention what we talked about briefly yesterday, that the subset of myofibroblast can, or a subset of the adipocytes can transdifferentiate to matrix secreting myofibroblasts and vice versa. So I guess the big thing was when one, as we mentioned yesterday, that one thought that myofibroblasts were terminally differentiated cells, but then a subset of the myofibroblasts can actually differentiate, de-differentiate or differentiate into, re-differentiate into adipocytes. And during wound healing, one has also seen that adipocytes can come into the wounds and form myofibroblasts. So in that case, in that pathological situation, it's really adipocytes that form the myofibroblasts. And as we said, myofibroblast after injury can disappear by apoptosis, so that you will not end up with a tissue filled with fibroblasts. But more and more one is seeing that that is not always the rule. So in the heart, for example, when you've had an injury, the fibroblasts do not undergo apoptosis, but they become revert to quiescent state, resting state, which is a state that is different from the original state. So it's not activated, but it's not the same resting state as before the injury. So we're constantly learning new things, and there are different mechanisms in every tissue. I think that's an important lesson. Here is a summary of different cytokine pathways that drive fibrosis. We talked about TGF-beta that's secreted by macrophages that can activate the fibroblast. We've talked about IL interleukins. Right, so then in the very last few minutes, we'll say a few words about CAFs, which you will hear more about. So a few years ago, or 10 years ago or so, much focus in cancer research was on the cancer cells, and also in fibrosis. The difference was not the focus was not so much on the fibroblast, but now all of a sudden there is a great interest in understanding the mesenchymal cells that we call myofibroblast in fibrosis, that we call CAFs in cancer, and how they contribute. So it's absolutely clear that they are important. Here shows CAFs in red, how they send signals into the tumor cells, how they can generate the enzymes that enable the tumor cells to metastasize. And we have summarized the role of CAFs, and specifically the integrin that we work on, in 3 different tumor forms here. In lung, the CAFs in an alpha-11 dependent manner seems to secrete different growth factors that influence the tumor cells. In the breast, we have, together with the Belgian group, shown that CAFs that express alpha-11 integrin collaborate with the TGF-beta receptor to stimulate signaling in CAFs, which then leads to secretion of tenascin C that affects the tumor cells. In the pancreas, there are no in vivo data, but it seems from a combination of in vitro and in vivo experiment, it seems that alpha-11 contributes to CAF activation in the pancreatic tumors. And this is another picture showing the role of CAFs with regard to chemoresistance, which is another important novel function of CAFs. One realizes that the signals and the contacts that the CAFs make are important in development of chemoresistance. Just a few super good articles that have been published the last year, showing here, summarizing several papers where one can differentiate fibroblast into 3 major populations: myofibroblastic CAF, inflammatory CAFs, and antigen-presenting CAFs, which are subsets of fibroblasts existing above all in pancreatic and breast cancer. This is where this has been characterized. And here are some of the genes that have been found associated with these subtypes. And here is another picture showing these 3 different subset of CAFs. And different signaling pathways support these different phenotypes. So in myofibroblastic CAFs is TGF-beta, in inflammatory is interleukin signaling. And here again is looking at different clusters of cells with different expression patterns. And here actually they compare mouse with human and find that there are differences, important differences between mouse and human fibroblasts. So things are much more complex than we previously thought. This is my last slide now, so I will not be over time. So the final thing that they do in this excellent paper is that they compare data, transcriptional data from different solid tumors and find that there is an 11 gene signature in myofibroblastic calves that's shared across tumor borders. And in that, for us, this was interesting because there is only one integrin in this 11 gene signature, and it's integrin alpha 11. So I think we skip this. These were towards the end, there were some examples of therapies aimed at targeting the stroma that have been tried, but largely unsuccessful so far. So we're still waiting for a tumor therapy that will target the stroma specifically. Okay, thank you. We have any questions?
Thank you very much, Donald, for very informative lectures. So please answer questions now. You can do that by raise hand, chat, or in other ways. Any questions for Donald? No, I can't. So if you have any questions, and some of you are also part of the course, you can always ask more questions during the group work or the lab presentations. But if not, I think we have a small break now until 9:30. Okay, here we have somebody. This is from Mamona. What are the functions of alpha SMA marker?
Alpha SMA is stands for alpha smooth muscle actin. So it's an actin isoform that is normally expressed in smooth muscle cells, but in activated fibroblast in a TGF-beta dependent manner become activated in fibroblast. And this actin form is more contractile, so it makes the cells more contractile. They can reorganize the matrix better.
Okay. No more questions. Then I think we take a break, approximately 12 minutes to 9:30 Norwegian time. And then we will come back and listen to Ritva about collagens. Okay?